Ding, there we go. Right, so Mendel comes up with those results that he's getting from his crosses, where in the F2 he keeps getting this 3-to-1 ratio, and now he's got to figure out why he's getting that. So let's have a look at why. He reasoned like this in a paper that he published in 18. There are little particle p's and we now call these elements genes. A gene can exist in several forms, and we call those forms alleles. From the Greek root allo means different. So alleles are different forms of a gene. We will get later into what exactly genes are, how they exist as physical entities, but don't worry about that yet. Mendel is still doing this essentially in his head. He doesn't yet have a way to see what genes physically are, although we'll start beginning on that actually on Wednesday. But whatever a gene is, it can exist in multiple forms called alleles, and one allele might be dominant or recessive to another allele. In all of the examples I've shown you and everything that you're going to see today, every gene has two different alleles. There's a gene that can either have a purple flower allele or a white flowered allele. There's another gene for P color that can come in the alleles green or yellow, and so on. In the real world, there are some genes that may have only a single allele, or there are genes that may have 3 alleles or 4 alleles. The human record that I'm aware of is a gene that's got 26 recognized alleles. I'm sure that's not the highest that's out there. That's the highest I'm aware of. And we'll get more later as to how to handle this. The genes that Mendel was working with just happened to only have 2 alleles within the experiments that he was doing. There's no reason that it has to be restricted to 2, but we'll keep it to 2 for today. And every pea plant must have 2 copies of each of its genes. So every plant got 1 gene from its mom, or the parental plant that contributed the eggs, and 1 copy of each gene from the plant that contributed the sperm. Every pea plant got 1 gene from mom and 1 gene from dad, and every pea plant will pass on 1 or the other, but not both, of every pair of genes that it's got. Those 2 copies might be of the same allele, or each could be different. Just like, if you're flipping a coin twice, you could get 2 heads, or you could get 2 tails, or you could get a head and a tail, or a tail and a head, of course. When a plant's 2 copies of a gene are both of the same allele, we'd call it homozygous. That would be analogous to 2 heads or 2 tails on a coin, 2 coin flips. If a plant's 2 copies of a gene are different alleles, we call that heterozygous. That would be like head tail or tail head. That's heterozygous. Zygo, by the way, means in Greek, that means a yolk. Not an egg yolk. I mean a yolk as in the thing that links a pair of horses or oxen together so they can both pull your plow. I don't know, there's at least a couple of music majors here, and if you've ever sung Handel's Messiah, you might know his yoke is easy, and his burden is light. His burden is light. Anyway, sorry. Yeah, that's a yoke, something that links together two draft animals to pull your plow or your carriage or your cart or whatever it might be. So zygo means linked together. Homo in Greek is same, and hetero means different. So homozygous just means same things linked together, and heterozygous is different things linked together. Okay. Any given allele might be dominant or recessive to another allele of the same gene. There's other possibilities, but let's not get sidetracked right now. Dominant, again, doesn't mean better. Like the flower color allele for purple does not beat up the flower color allele for white and take its lunch money or give it a swirly or anything like that. Purple flower color is not intrinsically superior to white or anything else. Dominant only means that a dominant allele will cover up the existence of a recessive allele. Dominant alleles will mask the existence of recessive alleles in an organism. So those F1 pea plants that we were looking at a few slides back must have had one dominant allele for purple flower color and one recessive allele for white flower color. They were heterozygous, right? They had one purple allele and one white allele, but they all looked purple because the purple allele was covering up the fact that they also had a white allele. So the purple allele was dominant and the white allele was recessive. And we can represent dominant and recessive alleles of the same gene by capital and lowercase forms of the same letter. The pros would actually do things a little bit differently. I'm not going to worry about that. This will do for what we need to do here. So we could represent a dominant flower color allele with a capital P, and the recessive flower color allele is represented by a lowercase p. And by the way, I can't really show it here on this slide, but if you're writing these down, because a capital P and a lowercase p look very much the same except for size, what I often recommend that people do is write their lowercase letters in cursive and their uppercase letters in print. You don't have to, but it may make it less. It may make you less likely to make errors just caused by misreading your own writing. So, okay, the reason we're not using P and W, by the way, is that we're representing dominant and recessive alleles of the same gene by capital and lowercase forms of the same letter. It doesn't really matter what the letter is. We could use A or B or Q or anything we wanted to. The professionals would actually use 2 or 3 letter abbreviations because they've got a lot more genes to keep track of. And they probably distinguish different alleles with little superscript numbers. Don't worry about that. Well, we have a gene for flower color, and we can represent the dominant purple allele with a capital letter and the recessive white allele with a lowercase of the same letter. I'll use big P and little p, but you'd get the same answer if you used big Q and little q, whatever. It's all good. So you can represent the alleles that any given plant is carrying by a pair of letters. A white flower plant has to have 2 copies of the recessive allele. And we can represent that with 2 lowercase letters, little p, little p. A purple flowered pea plant could be homozygous, which means it's got 2 of the dominant purple alleles, and we could write that capital P, capital P. Or a purple flower plant could be heterozygous and have 1 purple allele and 1 white allele. and we write that capital P, lowercase p. I usually just say big P, little p. It's easier to say. That would be a heterozygous plant that has the two alleles of the pair are different alleles. So Mendel's first cross looked like this. He's crossing a true breeding purple flowered pea with a true breeding white flowered pea. The P generation parents, one had two copies of the dominant allele, which we can write as capital P, and the other parent had two copies of the recessive white flower allele, which we can write as little p. Every one of the offspring in the F1 generation got one of the dominant alleles from one parent and one of the recessive alleles from the other parent. Every one of these plants got a big P from mom and a little p from dad. Or actually, it could be the other way around. It could be a big P from dad and a little p from mom. It's not important in this case. So every one of those F1 plants is heterozygous. They got a dominant allele from one parent and a recessive allele from the other. So we can write their combination of alleles as big P, little p. Ooh, it's computer animation. So that one got a big P from one parent and a little p from the other. That one got a big P from one parent and a little p from the other. That one got a big P from one and a little p from the other. That one got a big P from one. Okay, you get the idea. Here's what the F2 generation looks like. Each F1 plant is heterozygous. It's carrying a big P allele and a little p allele. And every plant passes on one or the other, but not both, of every allele that it's got. I'm saying, if I'm going slowly and if I'm making this sound like I think I'm talking to idiots, I promise you I don't think that. But bitter experience has taught me that this is something that not everybody manages to get the first time. But you're not going to get out of here without going through this. So let's keep marching on. Each of those F1 plants has a big P and a little p. Each one of those plants can pass on either its big P or its little p, but it doesn't pass on both, not normally anyway. And so there are four possible outcomes. Some of the F2 plants just happen to get a big P from mom and a little p from dad, and they end up homozygous for the dominant allele. They end up with two copies of that big P dominant purple flower allele. Some of those plants could get a big P from mom and a little p from dad. They're going to look purple, but they will be heterozygous. Some of these plants could get a big P from dad and a little p from mom, and they will be heterozygous as well. And then some of those plants will just happen to get a little p from mom and a little p from dad. And so there's four possible outcomes. You could be homozygous dominant. You could be heterozygous with one dominant allele from mom and a recessive from dad. You could be heterozygous with a recessive from mom and a dominant from dad. Or you could get two recessive alleles, one from each parent, and be homozygous recessive. All four of those outcomes have equal probability, and that explains why you get that 3-to-1 ratio. Getting a recessive allele from both parents is only going to happen one time out of four, for the same reason that if you flip two coins, you're only going to get two tails one time out of four. For exactly the same reason. It comes down to basic probability. To make this a little bit easier to visualize, we can use this calculating device that was invented after Mendel had died by a geneticist known as Reginald C. Punnett. He was British. He was at, now I can't remember if it was Oxford or Cambridge, but he was at one of the most prestigious British universities. played cricket with G.H. Hardy, one of the greatest mathematicians of all time, and developed this little, way of writing things down called a Punnett square. And it starts like this. Let's say we want to represent what happens if you cross a homozygous dominant purple flowered pea plant, big P, with a homozygous recessive white flowered pea plant, little p. Well, we'll start by drawing a square. That's a square. Ooh, ah. Each plant contributes one or the other of its pair of genes to every one of its offspring. So that purple flowered plant will pass on either one of its big P alleles or the other of its big P alleles, but not both. So at the top of the square, you write your two options, either a big P or another big P. The white flowered pea plant, you write in your options at the left. For the record, it doesn't really matter which one's at the top and which is at the left. The white flowered pea plant can only pass on a white allele, so it will pass on either one of its little p alleles or the other of its little p alleles. And then you draw lines in the square, and you fill the square in by writing the letters downwards and rightwards. So in the top left square, we write this. We take that big P from the top and move it down, and that little p from the left and move it to the right. What that's telling you is that 1 out of every 4 of these offspring will get a big P from one parent and a little p from the other. And if we fill in the rest, we get this and that and that. Okay. Possibly not terribly interesting, but this is what we'll get. What this is telling you is that 4 out of 4 of your offspring, which is all of them, are going to inherit one big P allele from one parent and one little p allele from the other parent. They'll all have the combination of alleles that we can abbreviate big P and little p. They'll have one dominant purple allele and one recessive white allele. And by the way, I think I forgot to define this term, but when you're describing what alleles a plant or any organism has, we call that the genotype. When you're describing what features the organism has, we call that a phenotype. So in the F1 generation, all of the offspring have the genotype big P, little p. They all have the phenotype of purple flowers. Okay, so genotype is a description of what alleles you have. Phenotype is a description of what you look like. So on test questions, if I ask you what is the genotype, the answer is going to be probably some combination of letters. So a genotype is going to look like big P, little p, or little p, or big P, big W, little w, or big O, big S, whatever it might be. It's going to be some combination of code letters. If the question that I ask you is what is the phenotype, the answer is going to be purple flowers, or green fruit, or flowers that start magenta and turn violet, or spotted coat, or sickle cell anemia, or inability to metabolize glucose properly. There's something like that. It will be some physical trait or feature of whatever living thing it is. That's a phenotype. Whereas a genotype is usually going to be some combination of letters that express what alleles we're talking about. So those are the possible genotypes of the offspring. 100% of the offspring got one P allele, big P allele from one parent and one little p allele from the other. So the genotype of all of the offspring is big P, little p. Since big P is dominant to little p, the phenotype of all of these offspring is purple flowers. Okay, that was not very interesting. This might be a little bit more. Let's set this up for the F2 generation. So at the top of our square, we'll put one of the parents, doesn't matter which, is carrying the combination of alleles big P, little p. That parent is heterozygous. To the left of our square, the other parent is also carrying big P, little p, right? That parent is also heterozygous. And if we want to crossbreed these, each parent can contribute either a dominant purple flower, big P allele, or each parent may contribute a recessive white flowered allele, abbreviated little p, right? So each parent can contribute either a big P or a little p, but not both. Now, we'll draw the lines in the square and fill it in down and across. 1/4 of your offspring will get a big P from mom and a big P from dad. They will be homozygous dominant. They will have one copy of the big P allele and another copy that is also of the big P allele. We can write their genotype as capital P, capital P. Half of your offspring, 2 out of these 4, will get a capital P from one parent and a lowercase p from the other parent. They'll be heterozygous. Their genotype is going to be big P, little p. Their phenotype is going to be purple flowers. Remember, phenotype is some trait or character or characteristic that we're looking for. And then 1/4 of the offspring, right? Remember, you're going to fill this in by moving those letters from the top and from the top of each column and the left of each row, from the top of each column down and from the left of each row to the right. 1/4 of your plants are going to get a little p recessive white flowered allele from each parent, and they will end up homozygous with 2 copies of the recessive white flowered allele. And because they have no dominant allele to hide that, they're going to look white. So the phenotype will be white flower color. So that's how you fill in that type of Punnett square. There we go, more computer animation for you. That's how you get that 3-to-1 ratio. This is what Mendel observed in his crossbreedings. This is what Mendel saw. What's more, 1 out of 4 plants will be homozygous purple with the genotype big P, big P. 2 out of 4 will be heterozygous purple, genotype big P, little p. Those are the ones that will look purple but can have white offspring if you let them self-fertilize in the next generation. And then 1 out of 4 will be homozygous white, genotype little p, little p. Ta-da! And there's our phenotypes. 3 out of 4 are going to look purple. 1 out of 4 will look white. And there's our genotypes using sophisticated computer animation. Nothing but the best for my students. Okay, right. All right, I tell you what. Go ahead, and this is actually a pretty good place to take a breather. I have 10:33. Let's reconvene at 10:50. I may let you clowns go early, but tomorrow we've got to put in an hour of working practice problems. So I'm going to go ahead and give you 15 minutes now. We'll probably finish early today, and I'll make it up to you tomorrow. We're not done for the day. I've got one more bit to talk about, to take a break and take a pause for a worthy cause. So I'm going to stop presenting. And there we are. I'm not seeing any questions on the chat, so I'm going to stop recording.